Hi dear students. Welcome to our last session on fungal diseases. In this session we will discuss about the laboratory diagnosis of fungal infections. In the previous sessions we have already discussed about few of the techniques that has been used for the detection of certain fungal diseases. According to Epstein and Purcell, there are few guidelines for the specimen collections, and it includes: the specimen should be collected from active lesion. Old burnout lesions don't contain viable organisms. And the specimen should be collected under aseptic conditions, that is without any other contaminations. You should always collect sufficient specimen. Use sterile collection devices and containers. And the specimen that is collected should be labeled appropriately. Methods of specimen collection include imprint. Imprint is a touched preparation in which tissue is touched on the slide and it leaves behind its imprint in the form of cells on glass slide. And the studies are made after proper staining. The other method includes swab, which can be throat swab or nasal swabs. The next one is the impression. Impression smears can be made from ulcerated or exudative superficial lesions, and the ulcerated areas should be imprinted before they are cleaned. The next method is the oral rinse, and the other method is the expectoration, that is to discharge matter from the throat or lungs by coughing or spitting. Then paper points and smears can be used as a mode of specimen collection. The specimens that can be collected include skin scraping, hair, nail, sputum, blood in the case of systemic mycosis, and cerebrospinal fluid in the case of cryptococcal meningitis. The fungal stains consist of wet preparation and differential stains, and the wet preparations mainly include KOH mount, Indian ink stain, necrosin stain, calcofluor white stain, lactophenol cotton blue, neutral red stain. And the differential stains include gram stain, Echani stain, Giemsa, PAS, Gomoris methamine stain, acridine orange stain, fluorescent antibody staining. In a wet mount, a drop of water is used to suspend the specimen between the slide and the cover slip, and this method will help prevent air bubbles from being trapped under the cover slip. Differential staining is a staining process which uses more than one chemical stain. Using multiple stains, it can better differentiate between different microorganisms or structures or the cellular components of a single organism. So beside direct microscopy, there is fungal culture, serological test, skin test, and PCR and other molecular methods for the diagnosis of fungal infections. Skin scrapings, nail clippings and hairs can be transported in an envelope, petri dish or other convenient conveyance. Specimens from mucous membrane may be directly inoculated in culture media or smeared on clean slide by swab or culture loop. Scrapings, crust, aspirated pus and tissue biopsies are to be collected aseptically from subcutaneous infections. In suspected systemic infections, specimens are to be collected from as many sites as possible, such as blood and cerebrospinal fluid. In KOH mount or potassium hydroxide mount, potassium hydroxide dissolves keratin and cellular material but does not affect fungi. Specimens are placed on a slide to which a drop of 10 to 20% potassium hydroxide is added and then covered by a cover slip and left for 20 minutes in incubator at 37 degrees Celsius to digest keratin, and the microscopical examination shows the fungal structure. The next type of stain is the Indian ink or necrosin stain. Because of its large polysaccharide capsule present in the fungi, it can be visualized by the Indian ink stain. Organisms that possess a polysaccharide capsule exhibit a halo around the cell against the black background created by the Indian ink. The particles of the ink pigment do not enter the capsule that surrounds this E cell, resulting in a zone of clearance or halo around the cells. The next one is the calcofluor white stain, and it is more sensitive and binds to chitin and cellulose of fungal cell wall, and they fluorescence under UV light. And this image shows a huge amount of aspergillus in cryptococcus stain with Indian ink, and yeast form of blastomyces shown with calcofluor white stain. Next is the lactophenol cotton blue. It is used to study the microscopic appearance of fungal isolates grown in culture. In this, the phenol will be acting as a disinfectant, lactic acid preserves the morphology of fungi, and glycerol prevents drying, and the cotton blue stains the fungal elements blue in color. And in differential staining, the first one is a gram staining method, and by gram staining, the yeast cells will be appearing as gram positive as it takes up the crystal violet, and it becomes more darker. For example, in the case of Candida, the hematoxylin and eosin staining of fungi show pink cytoplasm, blue nuclei, and no coloration of the wall. It does not disclose and distinguish fungal elements very easily. It is difficult to locate when the organism is in less number. The ginseng stain consists of the methylene blue and eosin stains, and it is used for visualizing the intracellular E cells, and it can be observed as purple nuclei, blue colored cytoplasm, and pink colored RBCs. The bar stain or the periodic acid skip stain consists of periodic acid solution as the skip reagent, and we can see the fungi as magenta, red or purple colored, and the background appears to be blue. The glycogen, mucin, and some basement membranes also appears to be red or purple in color. When Grocott Gomori methanamine silver stain or the GMS stain is used, the fungi histoplasma species appear black. The inner parts of the mycelium hyphae stained pink, red or rose in color. The mucin stains dark grey, and the background takes up the light green and stains pale green. When acridine orange is used, it has affinity for nucleic acids such as RNA or DNA, and the bacteria and yeast will appear bright orange in color, and the WPC appear pale apple green, and RBC may not stain or stain pale green. Fluorescent antibody staining is used to detect fungal antigen in clinical specimens such as pus, blood, cerebrospinal fluid, tissue sections where the organism is scanty. It detects fungal antigen in smear and section, and the antibody coated fungi can be demonstrated with fluorescence. And the fungal cultures include the basal media, nutrition division media, enriched and selective media, differential agar media, and the media for stimulation of ascospores, and the media that is used for biochemical test. The basal media includes the sebodextrose agar that is used for primary isolation, and the neutral SDA, and the SDA with antibiotics like cyclohexamide and chloramphenicol, gentamicin is added to avoid bacterial contamination. And in this you can see the colonies of various organisms in the SDA agar. The nutritional division media include the cornmeal agar, cornmeal green agar, rice starch agar. These are used to enhance the formation of chlamydospores. The enriched and selective media includes the blood agar that is used for the cultivation of Histoplasma capsulatum, blastomyces dermatitis, Cryptococcus neoformans, and the Candida will show a moist opaque colonies in blood agar. And the other one is the brain in heart infusion agar. And next one is the birdseed agar or the Niger seed agar. It is used for the primary isolation of Cryptococcus neoformans. On chocolate agar, the Candida appear as yellow-white colonies. In sunflower seed agar or Pals medium, Cryptococcus neoformans form light to dark brown colored colonies. In Sabacdos agar, the Aspergillus species form granular flat colonies with radial grooves. And the chrome agar is used to identify multiple species in a single specimen. In this, the fluorochromes are added and we can detect the enzymatic activity directly. The other one is the Biggie agar that consists of the bismuth sulfide, glucose and glycine. It is used for the isolation of Candida albicans, cruzi and tropicalis. The next one is a Pagano-Levin agar. It distinguishes Candida species based on their ability to reduce triphenyl tetrazolium chloride. In this, the albicans show cream to light pink color and Candida parapsilosis show red to maroon color, and also the tropicalis got this red to maroon color, and Candida cruzi shows white to cream spreading type of colonies. Media used for biochemical tests include tetrazolium reduction medium. In this, the tetrazolium is reduced in different gradients depending upon the organism. Candida albicans show pale pink color, tropicalis orange pink, parapsilosis rose pink, cruzi pink color, and Candida glabrata show pale pink. Another media for biochemical test is the carbohydrate assimilation media. Then there is urease test, which will determine the ability to produce urease enzyme. There is conversion of urea to ammonia, and the cryptococcus show positive urease and Candida species show negative to urease test. In rapid urease test, Christensen's urea-based medium is used, and there is conversion of urea to ammonia. There is conversion to yellow to purple color in the case of cryptococcus. Next one is the germ tube test. It is a screening test which is used to differentiate Candida albicans from other yeast. Germ tube formation was first reported by Reynolds and Broad in 1956. When Candida is grown in human or sheep serum at 37 degrees Celsius for 3 hours, they form the germ tubes which can be detected with the wet KOH films as filamentous outgrowth extending from yeast cells. It is positive for Candida albicans and Candida debris. Approximately 95 to 97% of Candida albicans isolated develop germ tubes when incubated in a proteinaceous media. The principle of germ tube test. Formation of germ tube is associated with increased synthesis of protein and ribonucleic acid. Germ tube solutions contain tryptic soy broth and fetal bovine serum, which are essential nutrients for protein synthesis. It is lyophilized for stability. Germ tube is one of the virulence factor of Candida albicans, and this is a rapid test for the presumptive identification of Candida albicans. The serological test used in medical mycology includes whole cell agglutination, lactase particle agglutination, passive hemagglutination, and also immunodiffusion is very widely used, counter immunoelectrophoresis, indirect fluorescent antibody detection, ELISA, that is enzyme-linked immunosorbent assay, radioimmunoassay, and in skin test it detects cell-mediated immunity and done in vivo and in vitro. It detects delayed hypersensitivity, and it is shown by occurrence of induration and erythema within 24 to 72 hours following intradermal inoculations of fungal antigens like histoplasmin in the case of histoplasmosis, candidin in candidiasis, blastomycin in blastomycosis, sporotrichin in sporotrichosis, and trichophytin in the case of dermatophytosis. Other methods include PCR, restriction fragment like polymorphism, protein electrophoresis, nucleic acid probes, serotyping, karyotyping. Accurate in situ microscopic appearance of fungal colony can be observed in slide culture technique. In this, a sterile slide is placed on a bent glass rod in sterile petri dish. 2 square agar blocks are placed on a slide, and the bits of fungal colony are inoculated onto margins of agar blocks. Cover slip is placed over our agar block. Incubate the petri dish at 25 degrees Celsius. Later, LPCB mounts are made and observed under the microscope.